With that, I would like to formally begin today's presentation and introduce Marcus Miller.
Hello, everybody. I'm Marcus Miller. I'm wildlife biologist at the West National Technology Support Center in Portland, Oregon, and we decided we needed to put this little webinar together for support of the Pocket Guide to Sagebrush, which we distributed several copies of here recently. And so this book was made possible by the NRCS, Bureau of Land Management, Fish and Wildlife Service, the Forest Service, the Intermountain West Joint Venture, Utah State University, Rocky Mountain Bird Observatory, and the Point Reyes Bird Observatory. In particular, efforts by Wendell Gilgert, Pat Shaver, Al Windward, and others brought this to be. And of course, our featured speaker and the lead author, Leila Shultz. And so she's going to talk a little bit about sagebrush taxonomy and sagebrush ecology. And with that, I'll give you Dr. Leila Shultz.
Thank you, Marcus. I am officially retired from Utah State University, but still maintain an office there. I'm a professor emeritus now, research professor emeritus. This project started in 2009. Started talking about the project. My work started several decades ago with sagebrush, but bringing the Pocket Guide to you started with a conversation with Pat Shaver and Wendell Gilgert at a 2009 sagebrush workshop in Utah. And since then, we've done state-by-state several sagebrush workshops sponsored by the NRCS. The intent of this guide, and we'll go to the next slide with the collaborators that have been involved, the intent of the Pocket Guide is to give you a tool that will help you with identification of different kinds of sagebrush in the field. It provides you with maps, detailed illustrations, photographs, and descriptions of the plant. Thanks to so many people that I've worked with through the years in a very large sense with Artemisia. I've worked with people on different continents looking at diversity within the genus as a whole, but it's the Western North American sagebrush that has been so important in land management, and I'm in the Department of Wildlife Resources, so it's a good place for me to be and collaborate with geographers and ecologists and geneticists at Utah State University. Heather Garrison was my student who's worked for her master's degree on the now infamous Bonneville sagebrush, and I'll be talking more about that. Okay, next slide. The Pocket Guide describes 27 different kinds of sagebrush. This is more than has been included in the Tridentata, the subgenus of Artemisia that we call sagebrush. That has 20 different kinds, including subspecies and varieties. The new classification on a monograph that I finished in 2009 added California sagebrush, sand sage, and an island endemic, Artemisia nesiotica, from the Channel Islands because of genetic work that shows the relationship within sagebrush. For the Pocket Guide, I've also included 2 of the hybrids that are widespread. That takes us up to 22, and then 5 more species that you could easily assume are part of the sagebrush complex. They are within the genus. They're closely related. That's the fringe sage, the fuzzy sage. Hetetyphoida is the birdfoot sage in Wyoming, and porteri, again, is a Wyoming restricted species, and bud sage. So 27 different kinds. In the Pocket Guide, we show the core distribution of sagebrush in the Intermountain area, but please know that with the sand sage, we go into the Great Plains and into Texas and Kansas and Nebraska, Oklahoma, and coastal California. Next slide. The classification, and I am a plant classifier, a taxonomist, the family for sagebrush is after ACE, the sunflower family, huge family of flowering plants. The tribe is the Anthemidae, which also includes western yarrow and pansies. The genus Artemisia has a lot of species in it. It's one of the biggest in the family, with about 400 species, most of which occur in Asia. In North America, we have 50 species. Within the sagebrush group, we've got these 27 different kinds, 20 species, and then a number of subspecies. Even though the greatest diversity occurs in Asia, the greatest biomass of sagebrush is Western North America. There is no shrub group that occupies as much acreage as sagebrush. Next slide. So in defining species and subspecies, we have a number of things in our tool bag. Morphology, obviously, is what you're going to use in the field to define the species, but in looking at what makes a good species or a coherent subspecies, we bring in genetics and ecology. And ecology is so important with sagebrush. Knowing the habitat that the plant grows in is very important, especially in defining subspecies of big sagebrush. And I think most of you who are dialing in have been in the field and are looking at big sagebrush in the field and probably struggling with the differences that you find between mountain big sage, for instance, and Wyoming sagebrush, and that's what I'm addressing today. We'll talk more about how you can tell those things apart. This photograph I took in Owens Valley, California, and mostly black sage in the view. Next slide. So the NRCS workshops have taken me into the field with people who are in land management. We have had a lot of good dialogues, and it helps me to understand what the management issues are, and the importance of some of the groups that we have, like the hybrid zone that we have, where mountain big sagebrush and Wyoming sagebrush come together, typified by this kind of habitat that Pat Shaver is standing in, and how that relates to wildlife management and ecological site classification. Next slide shows the area that we're overlooking in Wyoming, an area that had been badly eroded, and there has been restoration work in progress to stabilize the drainage. Down in areas where you have deep, well-drained sandy soils and more water, like along the banks of this drainage, you'll have basin big sagebrush. With Wyoming sagebrush in the drier sites and the upper elevations, typically you'll move into a mountain big sagebrush zone. Next slide. In northern Colorado, with Roger Rosentreter and a group of wildlife biologists in the field, we were looking at restoration of habitat for sage grouse and working to determine whether we had Wyoming sagebrush or mountain big sagebrush. And this is a characteristic morphology for Wyoming big sagebrush with a rounded crown, short flowering stalks. Next slide. Basin big sagebrush, as I mentioned, typically grows where the soils are well drained, the root systems are pretty deep, and it's taller than the other subspecies. It has a rounded crown and a wide flowering stalk. again, this is in California, Owens Valley. David Tart is in the photograph, Forest Service, one of the Forest Service people who's been important in this work. Next slide. And I'm standing with a stand of mountain big sagebrush in Washington. Typically, you have the short growth form, flat-topped crown with flowering stalks that rise above it in a kind of layer cake, birthday cake morphology. Looks like candles on a cake sticking above the crown. Next slide. The black sagebrush, I think some of you are finding some palatability preferences that are based on color form, and this slide shows two different color forms of black sagebrush, with the one with the dark straw, the straw-colored flower heads in the foreground, the grayer foliage in the plant in the center of the photograph. And Jeremy will talk more about palatability later in this presentation. Next slide. Silver sagebrush, Artemisia cana, and it's got several subspecies. It is easily distinguished from big sagebrush, which is tridentata, by the entire leaves. They aren't lobed except irregularly and only occasionally. Mostly the leaves are entire, and they're also deciduous. This is an important thing for you to know in terms of management. The leaves do drop off in the winter. They'll stay for a long time, but they'll turn yellow. This plant is already starting to turn yellow. The leaves are yellowing, and they'll drop off. Next slide. Low sagebrush is often confused with, or can be confused with a dwarfed, drought-stressed big sagebrush, Artemisia tridentata, sometimes confused with black sagebrush as well. And I'm trying to give you a close-up that will show the distinguishing characteristics. And one of the things that's important with the low sagebrush that will distinguish it from black sagebrush is that the flowering heads don't have stalks, and the bracts around the flowering heads are hairy. In addition, the leaves on the flowering stem, and you can see this on the right side of the image, the leaves fall off early. So typically, once flowering starts, you'll have a naked flowering stem, a leafless flowering stem. Very short, as you can see by the measuring stick. Black sage will have a stalk below the flowering head, as will the basin and mountain big sagebrush. Next slide. Okay, I am talking a lot about big sagebrush because this is the group that is most widespread, and I know in the field is the group that causes the most confusion. The left side of this image and the illustrations that are in your pocket guide are done by Linda Vrobic, an excellent botanist, field botanist, illustrator, and specialist in taxonomy. Did the illustrations for the guide and for my monograph. On the left side, you see what's characteristic of the flowering stalk of basin big sage. It's a broadly branched flowering head with long lateral side branches and many flowering heads on one stalk. The insert to the right of that is Wyoming big sagebrush that has shorter flowering stalks, shorter and narrower. And then in the middle you see subspecies Vasiana, the mountain big sagebrush, again with the short crown. The vegetative portion ends below the flowering stalk, And Wyomingensis, again, is shown on the right side of this. Wyomingensis tends to have an elongated central lobe on the leaf, and that's shown in the detailed illustration. Okay, now, fairly clean look at the different subspecies, and I'm going to talk a little bit about what happens when we get genetic exchange, introgression, which is gene flow, hybridization to one parent or another, and then back crossing. Next slide. The two types of leaves are going to be important when you're looking at species in the field, and in the measurements that I give in the field guide, you need to know the difference between the early ephemeral leaves and the persistent leaves that occur in the axis of the ephemeral leaves. The ephemeral leaves are like sucker shoots on a tree where you get elongation and kind of wildly variable leaf sizes and shapes. They elongate early in the spring and fall off. Physiologically, this is really an important adaptation for sagebrush. The physiological growth rates are higher. Rick Miller worked on the ecophysiology and showed the difference between growth rates in ephemeral and perennial leaves. And for identification purposes, look at those perennial leaves that reduced bundle of leaves called a fascicle that's in the axis of the ephemeral leaf. Okay, I think you've got that. Next slide. The flowers and seeds are minuscule. I think you all know that. This illustration, which is 3 millimeters in size, it's an electron micrograph I took showing 3 florets, each different floret by the different arrows, and the seeds are down below these united petals of sagebrush. And the seeds are at most a millimeter in length, so very tiny, many thousands produced per plant. The heads of the flowering heads have anywhere from 2 to 10 flowers in a head, depending on species and subspecies. Okay, next slide. The hairs on the leaves. The silver color that you see when you look at a leaf, that's coming from the hairs that cover the surface of the leaf, and they obscure the glandular hairs that are the site of synthesis for the aromatic compounds that are in sagebrush. But these hairs that give the silver color are very important. They're non-living cells. They're air-filled with stalks, but they produce this dense layer on the leaves that reflects light and heat and helps to reduce the heat load on the plant. So it's a very important regulatory mechanism. Next slide. Another adaptation that Artemisia leaves have, and differences between the species, is the amount of water storage tissue in the cell and the compactness of the photosynthetic layer. The first slide, upper left, is silver sagebrush, which is mesomorphic, grows in fairly wet sites, and the internal layer between the top and the upper and the lower epidermis, very thin layer of water storage tissue. The next slide shows big sagebrush, Artemisia tridentata, with a fairly thick layer of water storage tissue that transports water laterally across the leaf. And this is an unusual characteristic in plants, and part of the reason sagebrush does so well in dry habitats. In the extremely xeromorphic sagebrush, pygmy sage is an example. Artemisia rigida, the sagebrush in Oregon, and scabland sagebrush have fiber bundles inside the leaves. That pink dyed area is a bundle of fibers. It's kind of like a twig, and it's reduced the external layer on the plant, and that's a part of the water conservation adaptation. Next slide. Okay, two different kinds of roots in sagebrush are important to its whole strategy in growing. It's got tap roots and lateral roots, and the lateral roots extend out from the base of the plant and take the water from the upper surface. This is one of the reasons that other plants often don't get established between shrubs, because nutrients and water are being taken. The next slide shows a taproot that extends. The taproots on sagebrush extend very rapidly once germination happens. This is Roger Rosenkretter's hand holding a 2-year-old plant that he dug up. And you can see how quickly that taproot grows down. Sagebrush is does not have to have mycorrhizae in the soil, but it does much better if there are mycorrhizae. And in greenhouse experiments, the plants die out pretty quickly in the absence of mycorrhizae. So the lateral roots function in water uptake as well as redistribution. A very important paper in 2002 by Ron Ryle, Martin Caldwell, and Associates, published in Ecology, demonstrated that water is redistributed from low layers to upper layers. So in addition to water uptake, it's also being put back into the soil system. It's an unusual strategy and an important one. Okay, next slide. The glandular hairs that I talked about that are tucked beneath that layer of silver hairs, the little drawing at the base, and this is a slide that Durant MacArthur provided me. That drawing shows the glandular hairs. They're the living cells that are associated in all species of sagebrush. Artemisia pygmaea, the pygmy sage, is the only one that doesn't have a lot of these glandular hairs. It's got other strategies for preventing herbivory. So the terpenes, the lactones, the tar are bitter compounds that inhibit a lot of herbivory, but the coumarins that are in sagebrush also correlate with palatability, and as it turns out, the species that have a high coumarin content, like mountain big sagebrush and low sagebrush, are very palatable, important for wildlife habitat. Grouse, sage grouse feed exclusively on sagebrush. And the test that some of you may have heard about or tried in the field, I'd like to hear more from you about it, is whether the plant fluoresces in the presence of a black light. And to do this test, you crush some leaves, put it in a vial of water or alcohol. There'll be slight differences based on the medium, and use a small black light. You can actually buy these battery-powered black lights now, so the tests are getting easier for $5, and it works as a pen light. So in the dark, in a motel room at night or a campsite, you can do these tests and look to see if the plant, if the leaves are fluorescing with this bright blue color. On the left, you see the bright blue of mountain big sagebrush. The vial on the right is Wyoming big sagebrush that doesn't fluoresce. Protein in the plant is very important. The seeds have about 20% protein, and since the plants flower late in the summer, produce seeds in the fall, the winter, it becomes a very important for winter browse. Next slide. Habitat differentiation. In the field, very clearly you'll get bands of one species or another, and subspecies will be separating out in that way as well. Along drainages, this is a site in northern Utah, drainages along the moist area with willow. In the center of the photo, you get silver sagebrush, the sicky silver sage, Artemisia cana acidula. Upland sites in the foreground of this picture, you have the mountain sagebrush. And the band, there is a band in here where Artemisia cana and Tridentata hybridize. It's a narrow band. It's not widespread, but it is something that happens that you need to be aware of. Next slide. The hybridization that we find in the field is very rarely occurs between species, but it is common among subspecies. And mountain big sagebrush, Wyoming big sagebrush, basin big sagebrush are subspecies of Artemisia tridentata, and that's where you get a lot of complexity in morphology. Next species. Next slide, sorry. Bonneville sagebrush is one of those hybrids, a hybrid of Wyoming big sage and mountain big sagebrush, usually in a very narrow band. Next slide. In some studies with a lot of field sites and using a predictive model to expand from the sites that we field tested, we found very clear zonation. This is Rich County in northeastern Utah. We found very clear zonation bands with mountain big sagebrush at higher elevations, Mason at lower elevations, Wyoming on very dry sites, in the Bonneville hybrid in between. And the paper by Heather Garrison and Durant MacArthur and I is just now in press naming that hybrid. Next slide. Polyploidy gives an adaptive advantage to these plants. Within the Artemisia tridentata complex, most of the subspecies exist as diploids or polyploids, diploids with a pair of chromosomes, polyploids with a multiplication of that pair. Wyoming's big sagebrush is always tetraploid with a chromosome number of 36 as opposed to 18, the diploids. And the higher ploidy level gives the plant more genetic material, more flexibility in adapting to very dry sites. Next slide. The major divisions of Artemisia, I talked initially about there being 400 species. The Tridentata is one subgenus within Artemisia. The Eurasian sage is strictly in Europe and in China, Asia. But in North America, we have the wormwoods, the sagebrush, and Dracunculus. The subgenus Dracunculus is tarragon. Next slide. The molecular work that's been done is defining these subgenera, but very clearly, there's a bright blue thing there that I added here to show you that Seraphidium is a distinct group, and it is Asian. Those of you from Colorado might be using a flora that calls Artemisia tridentata a Seraphidium, but that has been discounted now. We can see clearly that the Asian group is distinct from the Tridentata, and that's why I put this slide in. The next slide. Next slide. All of the Artemisias in North America are treated in the Flora of North America treatment that is online. Lots of technical terms, but it gives you an idea it gives you keys to all of the species in North America. Next slide. The monograph that I put together, published in 2009, covers all of the species that are in the subgenus Tridentata, and brings in sand sage, the coastal sage, and the island sagebrush as part of that group. Next slide. In the pocket guide, the last page, we've got this diagram that places the different species and subspecies in the sagebrush complex in an ecological gradient, with the x-axis being dry to wet and or low elevation to high elevation, and the soil moisture gradient going from low to high. So the upper right corner is going to be the coldest, wettest site, where we get the snowfield sagebrush, Iciformis, and silver sagebrush. Next slide. Okay, my major points in this presentation, and I hope the take-home message that you get is that, and I know those of you with field experience know this to be true, that the species and subspecies occupy unique ecological niches. Hybridization and polyploidy have contributed to the adaptability of the Tridentata and a lot of the aspects of leaf anatomy. Structural aspects are important. Habitat disturbances, though, are stimulating have in the past stimulated diversification of species, and disturbances that we're seeing like wildfires and reseeding efforts, we're getting some different genotypes coming in at sometimes very large scale, and we might be seeing different levels of different kinds of morphology and back-crossing into the local population. I won't get into the wide-scale disturbance, but just to say, in places like Idaho, where sagebrush dies after a fire, the competition from non-native species, we're finding thresholds where sagebrush is not recovering. The work that you're doing with ecological site class descriptions is redefining and clarifying our understanding of the ecological niches that different species and subspecies occupy. I find our time in the field is very important, and we're learning a lot from what you're doing in classifications. And in conclusion, the next slide, just to let you know, I think with the pocket guide, after you've spent some time with it, you'll see from the descriptions of species and the distribution maps that the species are well-defined and relatively easy to identify. It's going to be at the level of subspecies that you'll have difficulty. The descriptions will help, understanding of the sites will help, and the ecological site descriptions that you're developing usually correlate with environmental gradients that go very well with the different species and subspecies. Jeremy is going to take over at this point, and Jeremy, however you want to handle Jeremy Maestas, is going to talk some about wildlife use and chemistry of the plant.
All right, thanks, Leila. Can we get the next slide, please? So good morning, everyone. This is Jeremy Maestas. I'm in a joint position with NRCS and the Intermountain West Joint Venture, helping to get our sage grouse initiative on the ground. So I'm working within agency and then across partnerships to help do that. And the idea for this webinar came about from Marcus, myself, Leila, Wendell Gilgert. We really felt it important to take and not only send out this publication that took many years to put together, but provide some follow-up to field staff and others who are actually going to use the document. And so I'm really, humbled to be up here with Dr. Shultz and And just want to offer a few kind of closing comments about how this relates to our collective efforts to help not only sage grouse, but the whole sagebrush ecosystem. Clearly what we're doing is not just for these critters, but it's a larger recognition that the system, this big gray system we work in, is really diverse and it's really complicated, and that deserves a little bit more respect. So I'm just going to tie in a few thoughts on how this relates specifically to kind of our wildlife habitat conservation and restoration efforts. Next slide, please. So anybody who's worked in the sagebrush system for any amount of time will undoubtedly hear from someone that, basically sagebrush is worthless because nothing will eat it, which clearly is not true, and there's a number of studies done that prove that's not the case, and obviously if you spend any time observing wildlife and livestock, both consume it to some extent, but it's really unfair to talk about sagebrush as one species. Clearly, with 27 different kinds just to recognize in this pocket guide, there's a lot of nuances to this, okay? So here's a slide that I adapted from Roger Rosentrader's 2004. All right, thank you. 2004 Forest Service publication, and what Roger tried to do here was basically identify a relative scale of palatability of the different species and subspecies of sagebrush. And so, a lot of this is based on observation and some study, but palatability is kind of related to kind of the taste of the plant and relates to this balance that herbivores have to make between nutritional content and the defensive chemistry of plants. And so this scale kind of shows the top, plants that are higher in those coumarins that Leila mentioned, and at the bottom, plants that are lower in coumarins. So just a relative scale of palatability, not really specific to any one animal, but in general, this is kind of how it plays out. You might notice here that even within subspecies, like we talked about mountain, Wyoming, and basin big sage, you've got mountain and Wyoming are relatively high on the palatability list, whereas basin's further down. So, even when you talk about palatability of something like big sagebrush, it can vary quite a bit. And really, this doesn't say anything about the preference. Preference depends a lot on individual animals and populations and what's available to them in their environment. Can be related to behavioral selection, and so this is simply just based on the plant chemistry. This is how palatability generally shakes out. And I always make a point of saying that it's not all about how palatable or what plants animals can eat. Of course, cover plays an extremely important component to any landscape for wildlife and animals in general. So even the least palatable species on this list should provide very important cover requirements. Next slide. And there's some seasonal variability in that palatability that can happen. So sagebrush has, throughout different times of the year, concentrations of those chemicals, those defensive compounds vary. The glands, in general, throughout the year, you've got in the spring higher levels of defensive compounds, and in the winter lower levels. So this is primarily due to that semi-evergreen nature of sagebrush, where you've got these persistent leaves that are produced in the spring that are really full of chemicals and make it difficult for animals to consume a lot of it. And Roger Rosentrader has a great quote that I stole here that basically says, the easiest way to think of it is in terms of humans eating chips and salsa. In the winter, sagebrush is just like eating a bunch of chips. You can keep eating them. In the spring, it's like eating chips with really, really hot salsa. You're just going to have to eat less. So that's a good analogy for how these compounds can vary throughout the year, and maybe explain why we see animals selecting different types of plants throughout the year to meet their forage requirements. Next slide. Here's a great study that's in press, due out in Ecology sometime this year, that really speaks specifically to sage grouse. And this is done by Graham Fry, Jack Connolly, Dave Musil, and Jen Forby out of Idaho. And basically what they were able to document for sage grouse, which are a true specialist in foraging and utilizing sagebrush, they documented there's this kind of multi-scale selection going on in terms of which individual plants that grouse actually eat. And so from the largest level being the habitat type down to the patch and then all the way to the individual plant, they were basically found that there was higher nutritional content and lower levels of what they call these plant secondary metabolites, which are those defensive compounds. And what was interesting here is that in Idaho, in their study sites, basically at the largest scale, sage grouse selected black sagebrush stands or black sagebrush habitat types over Wyoming big sagebrush to forage in. And black sage had a lower nutritional content, but it was so much lower in those defensive compounds than Wyoming big sage that it was apparently a good trade-off and worth it for the birds to consume more of a lower nutritional plant. But then at the patch scale and the individual plant scale, they definitely saw, that the birds selected particular patches and plants that were both higher and in nutritional content, like protein, and lower levels of defensive compounds. And so, some of the take-homes from this are that, basically the dietary constraints of herbivores and species like sage grouse limits their use of the landscape. So we may have really vast areas that you might ask yourself, do we really need all of that in sagebrush? And you kind of go, well, since we understand so little about what's going on at the chemical level where, animals are actually selecting individual plants based upon these trade-offs, it really speaks to the fact that, these are a landscape scale species that need a large buffet to choose from, because we simply don't know which patches or individuals that they're going to choose. And that if we only have a limited amount for them to select from, they're actually, their fitness may be affected by that. So next slide. Okay, moving on to kind of, habitat rehabilitation, revegetation. As managers, we're often faced with the difficult task of trying to fix areas that are broken, but that can be costly and carry a very high risk of failure, especially with seeding. And so I just pulled out one individual publication here from Mahalovic and MacArthur, 2004. When you're trying to put sagebrush back, there are some things you ought to consider, and some of these may seem trivial, but I assure you they're not widely implemented, and we still find that some of these things are ignored when seeding specifications are put together. But they put together a great set of plant seed and plant transfer guidelines, and offer some recommendations here that basically there's some transfer zones you ought to stay within when you're moving species around, and that basically they say it shouldn't be more. You shouldn't go more than 300 miles from the original source, and then in particular with the big sagebrush subspecies, considering these moisture and elevational gradients that Leila talked about is of the utmost importance. So basically, your mountain big sage you need to put in your higher elevation areas, your Wyoming on your dry sites, and then your basin in your alluvial bottoms and things like that. So there's some great information in there for you to follow up on and take a look at. one thing to keep in mind is that it's clearly more important to match the sagebrush species to the environment than to pick and choose sagebrush species or subspecies based on palatability to wildlife. So really you need that ecological site level information to understand, put the right species in the right place and things will sort themselves out from a palatability standpoint. A few other thoughts here. Basically, as Leila mentioned, the seeds are so small, you're not going to be able to distinguish one species from another when you get them. So, when you write your specifications for sagebrush seedings or plantings, you really need to specify very clearly subspecies, if you have it, species, and require this. If you're not getting it from a commercial site where they grow it out on transplants or something, you need to be using this source identified material where they actually, when they wildland collect it, they have a lot of information, a certified program they go through to make sure that it's, where it's coming from, the elevational band it was collected in, and you'll have some idea. But, with all of that, it still sometimes makes you wonder what we're getting, especially when we do the big sagebrush seeding. The pictures at the bottom here are a pretty clear example of, if you were out there with a tennis racket collecting seed and you were in the commercial seed business out there collecting, which one of these plants would you go after, right? The left-hand side is a spindly, old, dry-looking Wyoming big sagebrush plant, and the one on the right is basin big sagebrush, just falling over with seed, probably right along a road or a road cut. So, when you get paid by the pound, and these seeds are microscopic, we may have some issues yet to work through on, ensuring that we get the subspecies that we want. And Leila mentioned, even recommending getting a voucher specimen from the collection site to know for sure. But I think we've got a long way to go in recognizing that species and subspecies selection, putting things back in the right place, is probably a huge part of failures that we've had in the past and something we need to work on getting better in the future. Next slide, please. So just in closing here, to me this is all just a complex and humbling amount of information that speaks to why we talk about keeping the best of what we have left from being fragmented or lost. So, we really need to work to ensure that these large intact areas are kept that way, which is kind of our mantra with the Sage Grouse Initiative and a lot of our partners now. Is it's once Humpty Dumpty gets broken, we may not be able to put it back together again. So we really got to work hard to ensure that we keep these things on the landscapes and prevent damaging and irreversible thresholds from being crossed. So with that, I'll turn it back over to the moderator for questions.
John Tulin, you had a question? Perhaps you're on mute? Well, his audio does not seem to be working. He sent a note asking about the $5 black light.
This is Leila again. I just did a Google search and got several options. I'm sorry I don't have the maker of that, but I think you can find it pretty easily. If you don't, let me know. My email address I think will be available through this presentation.
And John says thank you. At this time, I am seeing no further questions.
Well, thanks to all of you. It's good to have so many participants.
Thank you to our presenters, and thank you so much for joining us today. This concludes our program. You may now disconnect.